So good afternoon, everyone, and welcome to this Stimulus Science in Action, another in our three-part series. This afternoon, my mind is on bright lights, because you are being hosted by the Faculty of Science and Technology here at the University of the West Indies, your place to shine. And we are featuring our collaborators at the Canadian Light Source, the brightest light in Canada. And your hosts are three of our bright young lights in physics. Mr. Orain Green. Orain, turn your camera on. He's there waving to you. Mr. Matthew Wilmot and Mr. Dudley Williams. Now, do not worry about diversity and inclusion, because last week we had a mixed student panel. This week we have all males, and next week we have all females. So do not worry, we are covering the inclusion of all genders and all people. And let me just tell you a few things about these young people. Mr. Orain Green is your main host. He's an undergraduate physics student in the Faculty of Science and Technology at the UWI, and he's majoring in biomedical instrumentation. He's also the Guild Council representative for the physics department. He's a UWI Stat ambassador and a member of the Physics Honor Society in the faculty. And if you guys want to know more about these programs, just ask Orain or contact me. Mr. Matthew Wilmot. Matthew, say hello, wave to everyone. Matthew is a student at Campion College. He just finished his CXC or CSEC examinations and is waiting on his results to go into sixth form. And Mr. Dudley Williams is a PhD candidate in the physics department at UWI Mona. His thesis is examining the effectiveness of radiation cooling for the regulation of indoor temperature in the Caribbean as a climate adaptation technology. I hand over to Mr. Orain Green.
Thank you. Good afternoon, everyone, and welcome to the second Stimulus Science in Action series, an event put on by the Faculty of Science and Technology at the University of the West Indies Mona campus, open to all. This series highlights the importance and unequivocal potential of STEM to solve environmental, health, and economic issues. I am Orain Green, and on behalf of my co-hosts Dudley and Matthew, I thank Dr. Singh Wilmot for such marvelous introductions. This week, we are thrilled to see the scientists at the Canadian Light Source in action. The CLS is located in the city of Saskatoon at the University of Saskatchewan and is one of just over 60 synchrotrons in the world. Synchrotrons are ring-shaped particle accelerators, or atom smashers, if you will. Synchrotrons are crucial for research in many fields, including but not limited to environmental, agricultural, forensic, and biomedical sciences. Without them, we would not be able to witness, much less comprehend, some of the governing principles of matter and its subatomic interactions. Synchrotrons are also fantastic exhibitions of what scientific collaboration can create. It is renowned, the CLS is renowned as the brightest light, sorry. The CLS is renowned as the brightest light in Canada. And I know by the end of this virtual visit, when we understand some more about the synchrotron light, we will all see why it is remarked as such a luminous beacon. Before I bring in Matthew to introduce the team from CLS, let me outline just a few rules of engagement for this session. First, I am asking everyone to ensure that their microphones are muted during the question and answer session. If you want to ask a question and speak directly to the CLS scientist, you raise your hand and you will be invited to unmute and speak after being acknowledged by me. You can even open or turn on your cameras if you wish. You can also use the chat feature to post questions and comments, and this can be done anytime during the engagement with the scientist or after. We do want to have an engaging session ready, so please get your questions ready. We have a couple of prizes to give away, so you will want to listen carefully and get ready for that. Now I invite Matthew to share some thoughts and introduce the scientists who will be in action today.
All right, thank you, Orain, and good afternoon, everyone. Like many of you, I follow the show Flash almost weekly, if you've heard of it. But because apart from the supernatural occurrences and unrealistic metahuman abilities that we see in the show, there is a strong basis in science. Yes, people, the particle accelerator, that big giant ring beneath the ground, which exploded, giving millions superpowers and including granting the Flash his signature ability to run faster than the speed of sound, is real. Synchrotrons and cyclotrons, two types of particle accelerators, are both explored in the show. And although a rupture is possible, it pains me to say that it won't give us all superpowers. But to show us what the synchrotron actually can do, we will see Dr. Toby Bond and Dr. Chitra Karunakarn in action on the beamline today. Dr. Toby Bond is a scientist in the Industrial Science Division at CLS. His specialization is in 3D X-ray imaging, which he has applied to sectors including energy, materials, mining, and agriculture. Toby holds a BSc and Master's in Science in Chemistry and is also an experienced programmer who works extensively with image processing, focusing on automation of 3D volume data analysis using machine learning and data mining techniques. An agricultural engineer by training, Dr. Chitra Karunakarn is the manager for Environmental and Earth Sciences Department at CLS and leads the CLS Plant Imaging and Innovation Research Program, promoting the innovative use of synchrotron technologies for agriculture and sciences research. Dr. Karunakarn has a bachelor's degree in agricultural engineering and a master's and doctor's of philosophy in biosystems engineering. Her research work has been published in 59 referee journal articles and 4 book chapters. Currently, she is an adjunct professor at the University of Saskatchewan and at the University of Manitoba, Canada. Toby and Dr. Karunakarn are supported by other members of the CLS team to deliver this stimulus today. I want to acknowledge the presence of all members of the team, especially Tracy Walker, Education Programs Lead in the Science Division, and Dr. Lucia Zwin, who is also a senior scientist at CLS. Now I now hand over to the team to take us over and show us what's in store for us today.
Hello, and thank you for that. My name is Tracy Walker, and as mentioned, I'm the education person on the team that's delivering to you today. I just wanted to take a minute to welcome you to the Canadian Light Source, as you can see our logo above here. I was just going to give you a minute to see what's around here. As you mentioned, we are located in Saskatoon, Saskatchewan, which also happens to be homeland of the Métis, and we share this land with the Nehiyawak, Nakota, Dakota, Lakota, and Anishinaabe peoples through a treaty numbered 6. So I just thought I would introduce a few things like that. So I'm going to switch my camera around in order to give you a view of what I see as I'm standing here on the mezzanine of our facility. It was mentioned that we are a particle accelerator that takes electrons, speeds them up to nearly the speed of light, adds an awful lot of energy to it in that middle ring there, so that when they come to the outer ring that you see here, and they go around those corners, they produce X-rays and infrared light in order for scientists to use in their research. And the research happens in the beam lines, and you can kind of see going under the windows there, there's sort of a straight section that's coming down there. So our ring is about 170 meters in circumference. We've got 25 beam lines that can all be running at the same time. But today, we're going to jump way to the end over there in that other corner to Toby Bond on the beam line. So I'm going to turn it over to Toby now. Okay.
Hi, everyone. My name's Toby, and like Tracy said, I'm a researcher here at the Canadian Light Source. So I'm one of the people who uses these beam lines to do science. So what we have here is the Biomedical Imaging beam line, which is a beam line where we use the X-rays that come off of the accelerator to basically take pictures and do 3D imaging of lots of different kinds of samples. So this was designed to do imaging with biomedical things like tissues, and other biological systems, plants, and things like that. But I tend to use it for looking at things like materials. And especially in my field of research, I mostly look at things like batteries, where we're trying to develop better batteries for applications like electric vehicles, where, to try and reduce greenhouse gas emissions and that type of thing. So the way that this works is, a synchrotron produces X-rays and it produces other kinds of light. And we can use X-rays to do lots of different things in science. So if you shine it on a sample, so I've got a couple of examples right here. You can either take pictures of it. You can use the X-ray to scatter off of it and figure out what kinds of patterns you get. You can look at what kinds of interactions it has with the material and get down to the molecular level, try and figure out what kind of chemistry is going on inside the material. And so on this beamline, we're really just taking pictures. And so we have this fancy synchrotron version of the same kind of medical X-ray that you would get in a hospital. But the big difference between getting a medical X-ray here and getting one in a hospital is that you would come here if you needed to take a picture of something really, really small, so it's kind of like an X-ray microscope. So I've got some samples here, and this is actually what I'm working on today. This is probably a little bit hard for you to see, but this is actually a small battery, a research scale battery that contains the kinds of materials we would use in an electric car. And so what we do is we put this, so I'd have a sample holder right here. I'd put it down here, and there's a beam that comes from a wall that's in front of me. If I'm standing right here, then the beam comes through me, and then behind me, there are a couple of cameras that are special cameras that are sensitive to X-rays. So normally X-rays aren't something you can see. When you go and you get an X-ray at the hospital, you don't actually see the beam of X-ray light, but it is there. And if you have a special camera, you can take a picture of it and actually see the X-rays as they go through you. And so different parts of me, my bones will cast a shadow on the camera behind me, but the skin won't cast much of a shadow. So you can use that to take pictures of different things inside of me. And in this case, I'm doing the same thing, and I'm just using one of these little batteries to take pictures at the microscopic level of what's going on. And so we can take pictures of lots of different stuff here. So there's lots of different kinds of researchers who come and use a facility like this. And the reason you would come to a synchrotron as opposed to just going and using a regular X-ray in a hospital would be if you either want to take pictures of something that are really small, or another big advantage to using a synchrotron is if you need to take a picture of something that's happening really, really fast. And the reason for that is if you want to take a picture of something in slow motion, you need to take lots and lots of pictures every second. And if you do that, you might have tried taking a picture with your phone in a room that doesn't have very much light. And when you try and do that, the picture doesn't look very good. It's very noisy, and it looks, you need lots of light to be able to take faster and faster pictures. So if you want to take those kinds of high-speed videos, you need a really, really bright light source. And it's the same kind of thing in this case. You basically need a really, really bright X-ray source. So X-ray is just a different form of light, and you need a really bright one if you're going to take pictures really quickly. So we can actually take pictures here at about 10,000 or 20,000 frames per second. So you could take a regular video and you could slow it down by a factor of about 1,000. So if you have something really fast going on and you want to see what's happening inside of it, a synchrotron is a really good place to bring to do that kind of imaging. So there are lots of different kinds of experiments that go on here. And I thought I'd just bring a couple of other examples of work that other people do. So this is an example of just some biological tissue. So you can see that you may not be able to see too closely, but there's some skin on the top here, and then some muscle tissue underneath it. This is the kind of sample that the beamline was designed for, and we can do lots of different kinds of imaging. But this is one of the places where we'd put a sample. So I've got this plastic tube here, and this is transparent, so we can kind of see what's going on. We'd put a sample in the middle here, and then if we want to take a regular X-ray, then I just close up this room and go outside, and then we control it from a computer outside in the other room, and then we just take some X-ray pictures, and that's very simple. But if we want to take a picture of something like this in 3 dimensions and actually have a full 3D model in the computer of what's going on inside of the sample, then what we need to do is we need to take a picture at lots of different angles. And so this is something called a CT scan. And we use these in hospitals all the time. Usually what we do is you'll lie on a table and they'll move an X-ray source and a camera around you and take a whole bunch of pictures at different angles. And once you have a picture of something from a whole bunch of different angles, you can reconstruct those. You can put them all together using a computer algorithm and then create a 3D model. And you have a perfect 3D representation. You can go inside and see what's happening at every single point inside of the sample or inside of you if you're getting a CT scan. The problem with doing something like that at a synchrotron is we can't really move the X-ray source because the X-ray source is a gigantic particle accelerator. So we'd have to pick the whole thing up and move it around, which wouldn't be very practical. So what we do instead is we have a sample sitting in the middle here, and we rotate it around. Sometimes we have very big samples, and so we actually have this entire area that I'm standing in, which is about 2 meters to either side of me. This whole thing actually rotates around. So if I was this was actually designed for imaging large animals like horses and cows. So if you had a horse, they would come in here, they would get the horse standing still right in the middle, and then we'd rotate this entire thing 360 degrees while an X-ray beam comes through here and we take pictures. Today, I'm just taking pictures of very tiny things. So I'm just using a very small little platform that rotates just kind of like this in front of me. But behind me, there are cameras that take pictures. The beam goes through here like a laser. And you know, the principle is the same no matter what kind of pictures you're taking. But we can design facilities like this to take pictures of all sorts of different things. So that's just one small example of the kind of work we can do at a facility like this. Taking pictures and taking 3D scans is something I do every day for my job, but we have over 250 people who work here and a lot of different scientists who do a lot of different kinds of experiments on all of the different facilities that we have. So hopefully this gives you a bit of an idea that, you know, anything that we can use light for and anything that we can use X-rays for, there's kind of a synchrotron version of it. So this is a good place to start to see that we can do the same things you can do with X-rays just using a synchrotron. And in some other cases, we can do things that that you have to have a synchrotron for and that are kind of more specialized. But this is probably something you're a bit more familiar with. So it can show you that even though we're dealing with something like a big particle accelerator, it's still, at the end of the day, just makes a beam, and we can use that beam to do regular science. So I think I'll leave it there, and we'll move on to Chithra next.
Okay, a few seconds to share my screen. For some reason, my computer is thinking, so just give it a minute. Okay. So for the students there, I want to show that I'm an agricultural engineer by training, but I want to give you all a flavor of what the facility can be used for different types of research. So I made the title, Invisible Systems Can Be Made Visible by the Synchrotron. So when we come back to the questions, we can ask you some questions, but basically these facilities, the synchrotrons, produce the main product, it's light and mainly X-rays. So you all will know X-rays. You cannot see, as Toby mentioned, but it can excite things, and you can look through using X-rays to study different systems, right? So for the students there is a lot of physics involved. As Tracy mentioned, it's a particle accelerator, so it's physics. I'm going to show you lots of applications in biology, so you will appreciate how much biology is being done in the facility. Sorry, there is another C for chemistry, but it's a lot of chemical, we call bulk information or spectroscopy. So we get lots of chemical information. But as you will see, appreciate that facility. So when you see that facility, it's a lot of engineering, right? So mechanical engineering, electrical engineering, computers engineering, and there is a lot of mathematics behind all these engineering systems. So just to give you a flavor of the students out there, it's all basically a lot of science involved. And as Toby mentioned, there are more than 250 people in the facility. They have different background. So what I'm going to show now is some applications in biology. So if you take, say, the sunlight, we all know sunlight is a common system, right? Sunlight is so bright. If you take the whole sunlight and put it in a 1 millimeter square, it's extremely bright. But the synchrotron is million times brighter than the sun. So you will appreciate what you can do with the brighter sun, right? So the synchrotron. Remember the example Toby mentioned, if you want to look at something in a dark room, it's very hard to look at it. But if you have a very bright room, if you use your cell phone, you can take images very nicely, very high quality images. Same concept in the synchrotron. If you go to a hospital X-ray machine versus the synchrotron, as the synchrotron is million times brighter than the sun, you can take images much better quality, also faster. So you will say, I will show you an example on baking bread. And scientists come to look at how the dough formation is happening in bread. And because the light is extremely bright, or the X-rays produced in the synchrotron is extremely bright, that's what people are able to collect data very easily. So this is can students find out what type of this crop is? If you have a biology background, I don't know whether it's common in Jamaica. It's a wheat crop. It's a major crop in Canada. So if you want to look through the system right now, if you take images with your cell phone, you are using visible light, right? Anything related to green, red, blue, it will be visible light. But what can you do with the X-rays? So I'm showing you 3 different varieties of wheat here. So what you see here is the 3 different varieties of wheat. But if you see those images when they are in the same developmental time or biological developmental time, it looks as if they are all in the seed stage. But when you look through them using X-rays, this is what you see. So one is still like a later stage of seed development, one is in between, and one is still in the flowering stage. So this is what I made, that things that whatever you cannot do with the visible light, you can do with the X-rays. And X-rays is one of the main products in the synchrotron. The next thing is some examples or applications. So this is a compound. You can take any flower, sunflower, gerbera flower. These are called compound flowers, right? So it is made up of more than one floret. And the question is, if it is a single flower or single plant, people know how the water and nutrients move in the plant. But the main question in this case is the researchers want to understand, when there are more than one flower, how the water and nutrient moves in the system. So they took this, the researchers from the university took this, this is the sample here you see here. They took that system to a laboratory X-ray instrument or a medical X-ray instrument. And this is the image they got, right? And then the same sample, when they took to the synchrotron, what do you see? You see much clearer images, also more details, internal details. This is the difference between a medical X-ray instrument or laboratory X-ray instrument versus the synchrotron. First, you get better quality images because the light is very bright. Also higher resolution. You can focus the beams to micron to nanoscale. So our hair is a millimeter, but if you go divide them by 1,000 times, it's micron, and you take a micron and divide 1,000 times, it's nanoscales, right? So very high resolution and very high detail. And the question is, so as Toby mentioned, you can flash an X-ray through your sample and get one image, which is a two-dimensional image, or you can rotate your sample and take at different angles, and you can do computer tomography or 3D, and then you get three-dimensional information. So the same flower we rotated, take three images at different angles, we put them together mathematically, and this is what you see here. It's called computer tomography or 3D images, right? And then it's not only it looks pretty pictures, but now you can ask the questions and you can virtually slice them at any plane. So you can virtually slice them this way or that way, and you can look at the internal details. So what you see is a cross section at the bottom, and you can see the flower is so symmetrical. So there is a lot of mathematics behind nature, right, that people are trying to replicate in artificial materials. And the question main here is the researchers are interested in the vascular system. So what you see here in red, green, and blue are different systems through which water and nutrients move. So the researchers can use this information to develop better crops for drought tolerance or disease resistant crops and different applications, right? So that's a nice one. The next example is, have you heard some citrus diseases? It may be happening in Jamaica too. So there is some bacterial diseases associated with citrus, and it's a big problem in the citrus industry all over the world. So the thing is, the bacteria enters the fruit and sits in the seed, and it is inside the seed. So there is no way researchers or like plant people don't know whether it's an infected seed or a good seed. And then when you plant that seed, when the plant takes like a few years to fruit, and then that time these oranges are infected with these bacteria, and it is not edible. And the researchers came to the synchrotron to look non-invasively, without cutting the seed, can you say whether it's infected or not? So we put them through X-rays, and what you see to your left is a normal seed, and to the right is an infected seed. So what you are seeing is panning through the sample, and you can see the infected seed has some holes or undeveloped cells. So this is another example with non-invasive, without cutting through your sample. You can look and understand what is happening in any systems. And then you can even also quantitatively find out the extent of the damage. So here is the blue area, which tells you how much is the damage. But for biological people, if you understand, you can see lots of these details in the seeds. What is the vascular system? So the idea that helps to understand how the bacteria is entering from the flower into the seed. So what is happening is this bacteria is coming along this vascular system through the water and nutrient moving channels in the plant, and that's how it's entering into the seed and making the seed infected. So that's just an example of X-ray images. But another application of X-ray images is exciting different elements in your sample. So you can tune the wavelength of the X-ray, right? Like in visible light, there is a red, green, blue, the main light has different wavelengths. Same thing in X-rays, you can tune the X-rays to different wavelength, and you can excite different elements. For example, here you are seeing in a leaf sample, you can have the sample and focus X-rays at different spot, scan the sample, and you can look at where manganese is present. So what you are seeing is manganese is here and zinc is here. This is a bigger beam, like 150 micron spot size, and you can focus the beam to different scales. And what you see when you go in smaller scale, 20 micron, you can start to see this manganese is present in particulates in the leaf, whereas the zinc is dissolved in water, so it's along the veins in the leaf. So this is another application. So you can excite different elements in your sample by tuning the wavelength of X-rays in the synchrotron. So that what you see here is the before leaf is micron. So this is micron scale resolution. And there are systems, and this is an example I wanted to show to students who are interested in instrumentation, engineering background. You can develop cool machines, right? We call them shiny machines. It's very nice. So you can have a sample here, like an electron microscope, and you can rotate them. But in addition to micron scale, you can go nanoscale. So what you see is the right hand side cross section of a stem from a lentil plant. But if you remember your biology class, you might have studied primary cell wall, secondary cell wall, middle lamella. So what you see here is the chemical information. The lignin is in red color, which is in the middle lamella, and cellulose and lignin are in the primary and secondary cell walls. So whatever we study, but we can visualize here, that's a very interesting aspect. Sometimes when I study, you see a lot of schematics in the books, but in the synchrotron, you can see live images. So that's a very interesting aspect. The last example is baking bread. Remember I said the synchrotron light is bright. That means you can take images very fast. So the question is, can you make a softer bread with less sodium or different gluten content? And the researchers wanted to look at the bubbles inside the bread dough, okay? But you can do that in a hospital x-ray machine. Remember I showed the flower where the hospital, the images are very, very not good quality. Also, it takes long time to take those images. That's the main reason the researchers come to the synchrotron, because they can take like 1,000 times faster shots of x-ray images. So then they can model these bubble formation. To take images like this in the laboratory or medical x-ray instrument, it takes 7 seconds. But what happens is these small bubbles becomes big, so they don't have a lot of data points to model it. That's the reason the researchers come all the way to the synchrotron. They put the dough in the machine, and then they take x-ray images, and then they collect these images, and what you see is the 3D images. Just a minute for some. Okay, so my computer is a little slow in responding. And what you see here is the same beamline that Toby showed you is for biological application. So this is a movie that the biggest animal they have done so far is a dog for prostate cancer. Let me see if the movie can play. Sorry, it's really slow. So what you see here, instead of like small samples Toby showed you, you have a big dog. So they want to image a particular organ of the dog for prostate cancer. So they have them animal, and they can rotate the animal to take x-ray images of a particular organ without sacrificing the animal. That's the real thing. Okay. Those are some things I wanted to show. So I'll stop here when the movie is done. So it's a cool place, but it's a lot of scientific knowledge behind it. So for students out there, science is amazing. You can do lots of cool things. That's the message I want to convey. Okay, we'll stop here. Do we want to ask the questions now, Toby?
All right. Thank you. All right. Thank you, everyone, for thank you for the presentation. All right. Now we'll open the floor to anyone who would like to ask any questions about the particle accelerator or the synchrotron. So I would invite my colleagues as well if they would like to ask any questions. All right, Matthew has a question. Go ahead, Matthew.
Okay, so my question, well, it's just a general question for everybody. So as you said, well, as we know, all the beam lines and all the radiation and light are obviously they all have to be contained, but in science we all know there is always going to be losses, there are always going to be errors. So I was wondering, are there any health concerns or any hazards concerning with a repeated exposure to these types of rays in just the general vicinity or in the labs themselves? Because they might leak, or you might, the same thing that happened to Marie Curie is basically what I'm asking.
So, hi. Yeah, they've spotlighted you now, Toby. That's okay. Hi, everybody. This is Tracy. In answer to that question, what I'm going to do is I'm going to back out of this hutch where Toby is setting up the experiment to show you how that is enclosed when he's ready to start his experiment. And you see this access panel, which right now tells me that everything is ready to go, except that the safety shutters, they are closed. And the only way that those safety shutters could open in order to let the X-rays come in is if Toby does a hutch lockup, which makes it very safe and closes these doors so that the scientists are not in the hutch with the X-rays at any point in time. So does that help answer your question?
Yes, it does. Thank you very much.
And I should mention too that the places where we do the experiments are actually made out of lead. So they're very, very thick, and they do lots of safety tests to make sure that when the beam is on inside this room, nothing outside the room will be exposed. And they do those tests once every year. They come back and just make sure nothing has changed. So there are lots of things in place to keep us safe.
All right, awesome.
These facilities produce different types of X-rays. We produce soft X-rays, also hard X-rays. So what Toby is doing, hard X-ray, meaning it can go from 30 kilo electron volt to 100 kilo electron volt. So it's a very hard X-ray which has much more penetrating power. But if you go to the soft X-ray beamline, soft means it cannot even penetrate a paper or depending on the energy of the X-ray. So depending on that, if it is a soft X-ray beamline, you may not see a lead edge because it has less penetration power and damage. Yeah.
All right. Thank you. Thank you, everyone. Again, if anyone has any questions, they can raise their hand or send a comment in the chat. I do have a question at this moment in time. So I recall that you spoke about the synchrotron light having 1,000,000 times brighter a light than the sun. So my question is, how hot can the temperatures inside the synchrotron actually get?
That's a very good question. Yeah. We might have to talk to our accelerator physicists to answer that one. It can get quite hot. So I can give you a bit of a sense. I don't know if I can give you a number, but on the beamline that we're using right now, there are lots of filters that we put in place so that by the time the beam gets out here, there isn't really much of it won't heat anything up. But if I go behind those optics, when the light just comes out of the accelerator, it's actually very, very hot. What it'll do is we actually have a filter that's made out of diamond, and that diamond filter will actually glow red hot when it's exposed to the beam. And so that diamond filter is actually put there so that it'll absorb all of that heat, and that everything that's downstream of that filter, everything that's after the filter, won't be exposed to the heat. So that's actually a very good question. On other beam lines, you won't get that kind of heat. It depends on what kinds of X-rays you're generating or what kinds of light you're generating. But generally speaking, that's something we do have to be a bit worried about. Oh, yeah, and one other thing Lucio is just reminding me, we also have to have a lot of different cooling systems to manage the heat. So there's a lot of engineering that goes on. In some cases, we'll have, it's mostly water cooling. So we'll have water pumping through some of the different optics. So we might use a certain kind of crystal, for example, to filter out a certain range of wavelengths of X-rays. And that crystal, if we don't cool it down with water constantly, it can actually damage the crystal. It can swell up from thermal expansion and crack. So that's actually, So we actually have to take a lot of care to make sure that we don't overheat the components because they're very expensive. And in fact, there are a lot of triggers in the facility that automatically shut the accelerator down if there's if the pressure, for example, in a water cooling system all of a sudden goes away and the water cooling isn't working, it'll automatically shut down the whole facility to make sure that we don't damage anything. So yeah, very good question. We actually have to do a lot of thinking. Well, the engineers have to do a lot of thinking about how to manage heat. People like me just use the beam once it comes through the shutters, but for other people, it's something we have to think about very carefully.
All right, great, that's awesome. This is truly a marvel of engineering. We have a question from Samoya. What type of specialized software is used to see the colored images of the X-ray on the leaves and the seeds, etc.?
So the color comes from exciting different elements, and you can give artificial color like to manganese, red, blue. So there are different types of software available, but the main thing is it's excited by the X-rays. So usually the images come black and white, but we give artificial colors to show different elements. When you shine X-ray, which is colorless, X-rays are colorless, right? When you shine excite different elements, and then we artificially give them, like if there is a particular element, for example, manganese present, they will show us bright white spots in the leaf, and then we give them an artificial color to show manganese is in red, copper is in blue, and zinc is in green. But actually, it's an artificial color.
Okay, so okay, great. All right, so I have another question. So
hold on.
Oh, okay, go
I
ahead,
have a
Dudley.
question
I didn't see your hand. Go ahead.
Sorry. So I wanted to ask about the energy consumption of such a building. What is called this entire facility? And the other question, what kind of battery technology did you show us in the beginning? Is it liquid metal or something else?
Sure. So I'll answer your first question first. It takes a lot of energy to run an accelerator like this. And most of that energy actually goes into powering electromagnets that control the path of the electrons. So we have to have very strong magnetic fields to keep those electrons going around a corner. I don't know what the exact power consumption is, but I know that our energy bill is it's in, I think, something like $2 or $3 million per year. Wow. I'm guessing a little bit, but it is quite a bit. So it's interesting, you know, I'd like to think that the research we do here, especially on things like energy materials and climate change related topics, hopefully offsets the energy that we use. There's not really a way around, using less energy in a facility like this. Although, to be honest, in the last 30 years, there's technology that's come available to make these facilities smaller. So compared to the facilities in the 70s and 80s, they are more efficient than they used to be. But nonetheless, it still requires quite a lot of power to run a place like this. And to your second question, so the batteries I was talking about are lithium ion batteries. So these are the kinds of batteries that you would find in your phone or your laptop. But we use the same kind of batteries in electric vehicles. Actually, I have an example of a battery here. This is the kind of cell that they would use, for example, in a Tesla electric car. They have about 5,000 of these in a battery pack, for example, in the Tesla Model S. But lithium ion is definitely the most common technology used for consumer electronics and for electric vehicles. So that's mostly what I work on. There are lots of new battery technologies that hopefully will be, more efficient and won't need rare metals like lithium and cobalt. So there are people who work on that type of thing as well. But for now, lithium is the only one that's kind of cost effective and can pack enough energy into a small space to work in kind of on an industrial scale. But there are people working on things like sodium ion batteries that are starting to show some promise, and metal air batteries. those all have challenges with them, that make them a little bit difficult to use in electric vehicles. But I'm hopeful that, as we do more and more research, there are lots of other people at this facility who do research on kind of new chemistries. The stuff I do is more focused on kind of current industry relevant stuff. So for the moment, I'm working on lithium ion, but I hope one day to be working on something that'll be hopefully a bit more sustainable.
Sustainable. Is it, when you look on the battery, do you see the electron moving as they charge or discharge? That's not what you look on.
In a way, you can. So the kind of imaging I do is more looking at what's happening. Kind of, it's like if you had a microscope and you're looking at kind of the microscope scale, but with an X-ray. So I can't see quite down to the level of individual molecules or anything.
Okay.
But we do have other kinds of techniques where you can actually watch changes happen due to the transfer of electrons. And so we might do something like spectroscopy that Chithra was talking about, where you can actually see a change in the chemistry that happens as a result of the electrons going back and forth. And that kind of work is really important if you want to understand, if you have a battery material and you want to know where the electrons are going in the material. So we do a lot of that kind of work here, although this kind of imaging wouldn't be how you would approach that type of problem. We would use another beamline to do that kind of work. So hopefully that answers your question a bit.
Okay, all right, great. So we have a question from Samoya right now. So Samoya, if you would like, you can turn on your camera as well and open your mic.
Good afternoon, everyone. I was asking about the roles in that facility. What are the roles of the different scientists? Last time we heard about lab tech, well, not lab techs, but the science officers. I don't exactly remember the name, but there are different roles in a lab facility, and we want to know like some of the different things that they carry out to allow everyone to carry out research.
Chithra, maybe I'll let you answer that one.
Sorry, Toby?
Oh, I was going to say, I'll let you answer that one.
I didn't get the question. Can you tell me, Toby, what the question is?
She was wondering about the different roles in the facility.
Okay.
So, we have scientists and engineers and different people who support the research. So she was wondering what kinds of different roles there are.
Okay, so we have more than 250 people here, and it's mainly divided into two main divisions. One is Accelerator division, another one is Science division, another one is Corporate Services. In Accelerator division, Machine division, there will be physicists who are in charge of this particular accelerator. There are engineers they have different engineers, electrical engineer, mechanical engineer, computer engineers. Because to build these beamlines and maintain it and operate, you have engineers and technicians, right? So they all come under the machine division. And then the science division, where we all belong, like myself, Lucia, Tracy, and Toby, we are scientists working and using these beamlines for research purpose. So we help people coming to do research, as well as sometimes we do our own work as well. And then there is corporate services. So you need to pay the bills, like electricity bill, accounting, managing these 250 people. It's a big facility. So you have human resources, corporate services, and systems technology, right? Lots of computers. So it's a big facility, and you will find that's a fascinating place. In this place, you can talk to different types of professionals at any day. If you like, for example, when Toby is doing the work and if some computer is not working, he has to call the systems technology. Or if a system is not mechanically doing, he may have to call a technician or mechanical engineer. So it's a collaborative work and the professionalism from different people that makes this facility very, very unique and very interesting place to work.
Okay, thank you.
All right, great. This is going so nicely. All right, so our next question comes from Bustamante. He asks do specific particles require more energy than others to excite?
So if I can, is that the elements that I showed in the leaf? So different elements have a different absorption energy. They can be excited with different energy X-rays. So for example, something like boron, we consider light elements. So you follow the periodic table. The starting, these are lighter elements, so they need very low energy X-rays. So you can excite boron with a low energy or soft X-ray, and then you can go higher energy. Aluminum and silica are also like medium energy, and then you can go like iron, zinc, very heavy elements. So the different types of elements, they follow the periodic table, and lighter elements we call soft X-ray or low energy, and then heavier elements like iron, zinc, arsenic, or cadmium, we consider them heavier elements. They need hard X-rays or high energy X-rays to excite them.
Okay, thank you. So we have another question from Seymour. So he says, good afternoon. And he said that very interesting presentations. So he asks, how can scientists in the Caribbean access this technology for their research? Can you give us an idea of the cost to do standard biosamples for diagnostic purposes? Say, for example, disease mutations, etc.
I think Lucia is the best. Lucia, do you want to take that, or I can answer?
I don't think Lucia wants to answer.
There are different ways Caribbean scientists can access the Canadian Light Source. So Canadian Light Source is a national facility, but it is open to any international researchers. So the easy method to come to the facility is you have to write a proposal, which is open twice a year, and it goes through a peer review system. So we call that general user proposal system. That's one way of access. For anything related to proprietary work or industries, that's where Toby comes from. Like we have an industrial science division group, where if it is a proprietary, you don't want to publish, and you have an industrial related work, you want to keep it confidential, you can go through the industrial science division. The third axis is collaborative. So right now we are working with Jamaica to get some access to you scientists out there to access Canadian Light Source. So there is a team of people from the Canadian Light Source and Jamaica working together, and that is another way we can help you to get some access to Canadian Light Source. That's where Lucia is having regular meeting with the Jamaican scientists, where you can get some access.
Right, Chitra, I'll just come in and add something. So there are some of us who are working with the team at CLS through Lucia and the team there from the Faculty of Science and Technology at UWI, with the help of the Ministry of Science and Technology, which facilitated this connection. And there are people from the Bodles Research Center where they're doing research in agriculture. There was a general meeting that was announced, and all of us who were interested attended. And then, there was a look at some of the abstracts and a selection of 2 projects, one of which comes from my research group and one from Bodles. So now the team is working with us. There will be other opportunities go forward, and Tracy and I, in fact, are also talking about opportunities through outreach for more of us, including our teachers, to learn some more and get involved in the programs at CLS. So we have a bright future looking ahead with the partnership with CLS and Jamaica and Caribbean scientists, in fact.
All right, great. So generally saying this feels so cool. So another question from Bustamante. He asks, are there particles that have a threshold for the amount of energy they can absorb before they become undetectable by the machine?
I can feel that one. So I'd say that basically every atom that you look at will, if you shine light on it at the right energy, you'll get one photon hits it and one photon will come off of it. And so we have detectors that can detect a certain amount. So if you have a very, very small amount of sample, it'll get to a point where we can't really detect it. You'll get to the point where there's just too much background noise and you won't be able to tell the signal coming from the atoms apart from just regular noise. The threshold varies a lot depending on what kind of elements you're looking at. So usually for heavier elements, it's easier to detect them. So for things like trace metals like arsenic, for example, we can look at concentrations that are in what we call the parts per billion. So one part, if you had one atom in 1 billion other atoms, we could detect it. But for other types of elements, it can be a lot more difficult. You need to have a lot more of it in order to actually be able to see it. So I hope I think that's what you're getting at. I hope that answers the question, but feel free to clarify it if not.
Yes, I think that answered their question well. Thank you again. So we have another question. This time from Harshan. They ask, are there any PhD opportunities in physics at the CLS? So how can we access more information and apply?
Go ahead, Toby.
Okay, sure. Well, so in most cases, we don't host PhD students directly at the CLS. We do sometimes host postdocs here, but most people get experience out of a facility like this by working with a researcher who does work at the CLS. So most of the people who do research here are academic researchers at a university, and they'll usually travel to the CLS to use it. So that's the way that I got experience in this facility. It was actually as an undergraduate. I was working as a summer student with a professor here in Saskatoon. And so I got to come over and collect some data as part of that. And then years later, when I applied for a job here, I had at least had some experience as a user, as a researcher. But at the moment, I think there are some staff here who do have some PhD students, but we don't really have a PhD program the same way that a university does. So usually there's kind of an indirect way there's lots of ways to get experience working here, but we don't operate, we're kind of part of the university here, but we don't, you wouldn't come to the CLS to do a PhD in synchrotron science exactly. You would be doing research in some other field, and you would use a facility like ours to do that research, is usually how it goes.
So Toby, let me jump in again and invite Harshan, let me invite you to come to the Faculty of Science and Technology, look at the opportunities for PhDs in our faculty, and then we connect with the CSL to get you there to get your to get some data. I think that's the way out for you.
All right, we're on a great momentum right now. So next we have, so Bustamante says thank you for your answers. So Anuka asks, good afternoon. What sort of materials or metals make up the synchrotron itself? I know you mentioned lead before for the walls, but they would like to know what makes up the synchrotron itself. Hello? Yes.
So yeah, there's a lot of different materials. There's a lot of concrete to start off with. When they actually made this facility to build the ring, they had to pour all of the concrete, which is a giant tunnel that goes around a ring that's it's about 270 meters in circumference. And so they had to pour that all at once. They had to pour a section at a time until they made it all the way around the ring. And so it was one continuous pour 24 hours a day. Then on top of that concrete, they have to line that all with lead to make the radiation shielding. And then aside from that, a lot of the other materials we use to build a facility like this, there's a lot of steel because there's a lot of plumbing and piping. We actually have a liquid nitrogen plumbing system that runs throughout the entire facility. So we have to have insulated steel pipes that go all throughout the facility because we use liquid nitrogen for cooling certain devices. We use it in our experiments, so we actually have little liquid nitrogen fountains around the facility. When you come here, you'll see a lot of steel pipes a lot of and then aside from that, I'd say wires. There's a lot, tons and tons of cable here to connect all the different computers, all the different detectors, instruments that control the motors. There's just a ton of wires here. Pumps. Oh yeah, and pumps too. We have a lot of pumps to move those fluids around, move the water and the liquid nitrogen, liquid helium, and that kind of thing.
Toby, if I could just jump in right now.
Oh, and for vacuum too. Oh, sure. Go ahead, Tracy.
So I've moved away from where they are, and I'm now outside the storage ring wall. This is the spot where I was standing when I talked to you at the beginning of the call, was up in that corner. And so coming down here, this is the concrete wall that Toby was talking about. And here's my hand for reference. And this is the door. So that's the width of the walls that is shielding the synchrotron from us. This is a spot where we could put a new beam line when we're ready to. And you can see the radiation monitors that are around and here like this. So there's no beam line here yet, but you can see that there's lots of equipment stored up on top of the ring, computer closets, and the vacuum systems and the air exchange system. And Toby also mentioned some cryogenics. So I'm going to walk over to, sorry, my video is choppy. I don't walk very smoothly.
That's okay.
This is one of the cryogenic tanks with the liquid nitrogen that's being fed into a different beam line. This is the outside of a different beam line that is just being locked up for another experiment. So that gives you some sense.
Okay. All right. Thank you. I see that everyone is avidly enjoying this session. I see we have another question from Matthew. You can go ahead, Matthew.
All right, thanks, Irene. So my question is, so when in the storage ring, after the electrons come out of the acceleration ring and they spin around for 24 hours, almost the speed of light, for maintenance purposes, do you have to take the electrons or take them out of the storage ring? And if so, how do you stop their acceleration or store them possibly? Yeah, something along those lines.
Chithra, do you want to field that one?
Yeah, so that's a good question. So the storage ring is an endless loop. So the electrons go around when they are accelerated. But when you want to stop, it's a particle accelerator, right? So you can bring a metal plate to bombard them. So it is a huge metal plate that they bring it in when they want to stop the operation. So we run like 6 days continuously, and every Monday there is a maintenance where they shut down the beam. So at that time they bring this metal plate to stop the electrons. The electrons go and bombard, and then basically they all die happily.
Oh. Okay, that answers my question. Thanks.
All right, great. I can see here that someone was also asking, give me a moment. Okay, Reason was asking, so they say that they have an interest in forensics. Does the CLS conduct forensic research?
Yeah, we do. Or we have had researchers who come here and have done, I guess, so, a lot of people who do research are chemists, physicists, biologists, kind of the classical scientists, but we also have a lot of people from anthropology, archaeology, art history. So those are all, forensic related fields, certainly, where you're trying to reconstruct something that happened based on, something that happened long ago, and you have some sort of artifact or object that that you want to investigate. So people have looked at, someone brought in a skull from the Bronze Age that had, I think it had an arrowhead in it. People have done research, not at this facility, but at another synchrotron. You can actually use a synchrotron to look through the layers of a painting. So something that they used to do a long time ago when painting canvases were really expensive is if you had a painting and you didn't really like it, you want to preserve that expensive canvas, so you just paint a new painting over top of it. And there's a research team in France actually that used a synchrotron to look at the paint that was underneath the painting. So there's two paintings on top of each other, and you could actually reconstruct the painting underneath the newer painting by zeroing and fine-tuning the synchrotron beam to the elements that are inside the different colors of paint, because they use different, they might use copper to make green paint or cobalt to make blue paint. And so if you can look at the distribution of copper and cobalt, you can figure out where the paint is supposed to be blue and where it's supposed to be green. So those kinds of forensic studies happen all the time at synchrotrons, and it's definitely a field of research that these are tools that anyone can use for lots of different kinds of research. So forensics and those kinds of applications are definitely something that we see a lot of.
Okay, great. So we have our next question from Janelle. She asks, is there a place for medical physicists in facilities like the CLS?
So
absolutely.
What do you see?
Go ahead, Jithra.
You are the right place. You are in the biomedical beamline, so you tell the story.
Sure. So I work with a couple of people who have a background in medical physics. So especially if on a beamline like this would be mostly where you would end up working if you were a scientist. So when we do experiments with live animals, for example, it's really important to understand the radiation dose. If you're doing any kind of medical imaging, that's where a lot of medical physics people would end up working. So you need to have that kind of background, which I don't have. I work on materials, and it doesn't matter to me if they get lots of X-ray exposure. But if you're working with a live animal, you need to be very careful about how much exposure you give to the animal. And so that would be one place where someone with medical physics training might work. Another place where we have medical physics experts would be in our health and safety department. So people who, like our radiation safety officer, for example, would have a medical physics background. That's less focusing on the research itself, but you're kind of working in the operations of the facility. But in both cases, those are kind of the two areas that come to mind when you're talking about medical physics.
Okay, thank you. All right, so we have here that Dudley says liquid nitrogen fountain, so cool. And Anuka is thanking you guys again. So Dudley's asking, is this facility below the surface? Like, is the synchrotron below ground?
So the accelerator is below ground, but the synchrotron itself and the rings and the beamlines are not. They're on the surface.
Okay, all right, thank you. Okay, so I'm opening the floor to any more questions. I do have a question, so I'm going to use the opportunity to ask mine. So I know that we spoke about the particle accelerator in shows like The Flash and so forth. So what fantastical aspects of the particle accelerator would you think they may have drawn inspiration from to utilize such utilize the particle accelerator in the show itself?
That's an interesting question. I think that there's a lot of mystery around. People. You'd have to work really hard to actually get exposed to the particle beam itself. For example, if we had something like an explosion here or a loss of power, what would happen is we have to work very hard to keep those electrons going around in a circle. And we have to have magnetic fields generated by electromagnets that are very strong to keep them turning around the corner. And the second you turn those fields off, they would basically just crash into the side of the steel pipe in the accelerator, and that would be it. So the kind of scenario where you have some sort of explosion that exposes the whole population to some kind of radiation isn't really going to happen at a particle accelerator. But there's one interesting point that I'd like to point out is that there's very, very little evidence for what happens. There's very little experience with people actually getting exposed to a particle accelerator beam. And in fact, there's actually only one person who's ever been exposed It was a Soviet scientist back in, the 40s, I think it was.
Yeah.
Was it 70s? So he's the only person who ever was exposed to a particle accelerator beam. He was working on an accelerator, and this was before we had good health and safety measures. He was working on an accelerator, and he had his head, he thought the accelerator was off, and he had his head in the path of the particle beam, and then they turned it on without telling him. So he actually got a particle accelerator dose to his head, and his head swelled up quite a bit. And he was I think he was blind in one eye, but he actually survived. And he lived to be, I think, 70 or like to an old age. But he was actually a really interesting experiment because nobody knew before him what would actually happen if a human was exposed to an accelerator beam. So that's the only case that's ever happened in the entire history of particle accelerators. So I think to maybe get back to your question, I think there's a lot of mystery around that because it's so rare. So you can imagine lots of different things that might happen if you're exposed, but we only have one example of when it actually did.
Okay, great. At least we have that one example, so we won't have to do it again. Yeah. So Roberto is asking a question. He says, "Great presentation. Does the accelerator allow for interactions with different quantum realms or the fourth dimension, if possible?"
I wouldn't say so. The kind of work that happens in a place like this is actually pretty routine. There's a lot of interesting quantum mechanics that go on, but we use it to do very tangible, everyday type of stuff. So we're not really doing anything in any extra dimensions or quantum realms that isn't outside of the existing sphere of knowledge, I would say.
All right. Thank you. Thank you. All right. So I'm opening the floor to any more questions at this moment in time.
Hi, Owen. I
Dudley?
think I have a question.
All right.
Yes, I think I have a question. Did I forget the question? Wow. Come on. So so what's the difference? It's called a atom smasher. Am I using the term right for the synchrotron? Because I'm wondering if you are actually smashing atoms together since you're just using X-ray.
You are correct that we are not smashing anything. In our case, we just run around in circles and then use the energy that comes off of that in order to do the research. The facilities like CERN, they do smash particles together in order to investigate what comes out of that.
Okay. Have you noticed any quantum entanglement with any other particles?
Sorry, could you say that again?
I was saying, have you observed any quantum entanglement?
So the kinds of physics that we deal with are mostly, we don't really deal with systems that involve quantum entanglement. The engineering physics that go on here mostly involve just trying to keep a really stable particle beam or a stable wave function inside the accelerator. Okay. There's a lot of really advanced quantum physics going on there. But in that case, it's a very carefully controlled environment, and it's a very isolated environment. It happens in a vacuum, away from any other kinds of systems. So something like entanglement isn't really something that our physicists would deal with. Potentially on some of the experiments that they run, where they do things like spectroscopy, and they're looking at things like superconductors and quantum materials like that, then you could be dealing with systems where that might involve something like entanglement, but the operation of the accelerator isn't really where you'd see something like that.
Okay, thank you.
Okay. Right. We are going to need to wind down now, so we want to get the prizes going. Just 2 questions, get the prizes going, and then we close. Okay.
All right. So if you may, you can provide us with 2 questions relating to the discussion to ask the students so that they may put their answers in the chat so that we may give them a prize.
Okay, so how is it going to happen? Is Toby, you have a question, do you? Did you find one?
Sure.
Yeah.
Yeah, I can start.
So once you drop that question, guys, the clock has started, and the first person to put that answer in the chat will win a Faculty of Science and Technology coffee cup. And we'll just need to get your details so we can send that on over to you. All right, Toby.
Okay. So what is one reason that you might want to come and take an X-ray image at a synchrotron as opposed to some other source like a hospital X-ray?
Ooh, all right, guys. Type as fast as you can.
There are a couple possibilities, but gotta just give one.
Okay, so we're asking the team to judge.
Okay, so Janelle says capture fast-moving reactions.
Yeah, that's a good one.
That's good enough? All right,
That's good enough,
congratulations,
yeah.
Janelle.
That is definitely
All
a
right.
reason.
All right,
Okay.
Janelle, reach out to me or Dr. Singh afterwards to collect your prize. All right, we can take one more question from you guys to the students to give someone else a chance to receive a prize.
Chitra, you want to do that?
Yeah, okay, maybe. So say you have a sample with different composition of different elements in your sample. How do you find or how do you excite those elements?
All right, let's see who
What
will be
are-
first. Let's see who's been paying attention. All right, haven't seen anything yet.
Did you get the question, Aaron, or you want me to repeat it?
You can go ahead and repeat it. Oh wait, we have one answer. So Elena says, shine a beam of light through them. Will you accept that answer?
Maybe a little more specific. I say a sample, like an example of a leaf has different elements. But how do you excite the different elements? What do you have to do?
What do you have to do to the light? Let me see if I can help out. What do you have to do to the light, guys? Say, for example, we're using magnesium. We're searching for magnesium or zinc. Let's see. Come on, guys. I know you got this. So Harshan says use different frequencies. So
Yeah,
will you take that one?
Yeah, frequency or wavelength. Yeah, that's
all
right.
right. Congratulations, Harshan. You can reach out to either myself or Dr. Singh afterwards to collect your prize. So at this moment in time, I'll hand over to Dudley so he can do our final remarks. Thank you again for your time, everyone. I really do appreciate it. The students really do appreciate it too. They've had a great time so far. Dudley?
Thanks, Owen. I am so excited. I can't even express how my mind is blown at this physics that we have learned today. First, I would like to thank the entire team at CLS, Toby Bond and Dr. Karun Nakaran for being in action at the Biomedical Imaging Beamline. I also want to thank Tracy Walker and Dr. Lucia for facilitating and organizing this engagement, and to all others who made this possible at the CLS, we say thank you. Something that resonated with me today, the power of a push. Here we saw that a tiny electron, given the right push, a very expensive one at that, was able to do extraordinary things. My audience, you are that electron today. Keep moving towards your dream. If you need any assistance, to stay in action, I would like to introduce you to the Faculty of Science team. So first, I would like to thank Dr. Marvadine Singh-Wilmot, the Associate Dean for Student Exchange in the Faculty of Science and Technology, for conceiving and organizing this event and working with us to deliver it to you. We also want to thank Terry and Colleen Frey from the Faculty of Science and Technology for organizational support, and Claudia Lewis for preparing the attractive flyer that advertised the event. We also thank the Faculty of Science for providing the prizes today, and our prize winners, we congratulate you for your participation. We also like to thank all those who also publicly publicized our event and for all those persons that came and participated. Before you go, please remember to give us your feedback by completing the short survey posted in the chat. Remember to join us again next week when we see scientists in action at the earthquake unit in the Faculty of Science and Technology at UA in Jamaica. So look out for the flyer or visit our faculty website and subscribe to our YouTube channel to keep abreast of all that is going on. The links are posted in the chat, and as you remember, the UA is the place to shine. And the Faculty of Science and Technology is your place to go for knowledge, research, and solution. Our faculty offers diploma, Bachelor of Science, graduate certificate, and diplomas, Master of Science, Master of Philosophy, Doctor of Philosophy programs in a wide range of fields, including physics, which is fun, mathematics is also fun, biochemistry, biotechnology, life sciences, chemistry, computer science, geography, and geology. So come and join the Faculty of Greatness. With that, I wanted to say thank you for your participation today.
Great. Let us all give a round of applause to everybody today, including these three gentlemen who delivered this program to us. You young people are fantastic, and I think it shows that Jamaica is in great hands. There were so many questions and so many comments that kept the team on their feet. I just want to also add my appreciation to the team at CLS. And Jamaica is looking forward. Jamaica and the entire Caribbean is looking forward to our collaboration with you and to having some of you come over here as soon as the COVID-19 is over, as well as those of us who are successful in our bid for proposals coming over there. So we're looking forward to some stronger interactions, everybody. And we will be seeing each other and some more. And I'll see you all next week right here when we go to the earthquake unit. Bye, everybody. Please keep safe. Bye, Lucia. Bye, Tracy. Bye, Toby. Bye, Chitra.
Right.
Bye now. Thanks for having us.
Thank you.